In the last video, I described how to make cDNA. If we want to clone a particular cDNA from a mixture, it is helpful to make many copies of that specific cDNA so that it is the most abundant species in the mixture. We do that using the polymerase chain reaction, or PCR. PCR is a technique that allows amplification, or making many copies, of very small amounts of starting DNA. PCR can be successful with as little as a single copy of the target DNA in the starting solution. PCR was invented by Kary Mullis in 1985, and since then people have developed many applications of the technique. I'll mention a few applications other than amplifying cDNA at the end of this video. But first I need to explain how PCR works. The double-stranded starting DNA, or template DNA, is represented here, with the target region to be amplified in the darker colors. For our cDNA amplification, the dark region would be essentially the entire cDNA molecule. The reaction mixture also contains two primers, which are complementary to the template strands at either end of the region to be amplified. Thus, you need to know the sequences at the ends of the region to be amplified. For our cDNA amplification, we can get these sequences from the published human genome sequence. For PCR, you must include a DNA polymerase to synthesize new DNA, and that polymerase should be thermostable, meaning it is able to retain its function even if heated to high temperatures. You must also include deoxynucleoside triphosphates as substrates for the DNA polymerase, and, as for any biochemical reaction, an appropriate buffer to provide conditions under which the polymerase will be active. To perform PCR, you change the temperature of the solution in 3 stages, and then repeat those changes for many cycles. In the first stage, you raise the temperature very high, usually to around 95 degrees Celsius, which should cause all of the DNA in solution to denature, or separate into 2 strands. Then you cool the DNA to a moderate temperature that will allow the primers to anneal specifically to their complementary sequences. Note that one primer anneals to one template strand, and the other primer anneals to the other template strand. The 3' ends of both primers are toward the region that is to be amplified. In the third stage, you raise the temperature to the optimal temperature for activity of the DNA polymerase, which is usually around 72 degrees Celsius for the enzymes currently in use. During this stage, DNA polymerase will add nucleotides to the 3' ends of the primers, resulting in duplication of the DNA in the region to be amplified. To make these changes in temperature easy, programmable thermocyclers have been invented that automatically raise and lower the temperature as desired. After giving polymerase enough time to synthesize the target region, you move to the second cycle of PCR and repeat the 3 temperature stages. You heat to 95 degrees to denature the DNA, cool to allow the primers to anneal, and then heat to the optimal temperature for the DNA polymerase. Note that the DNA strands that were synthesized in the first cycle become templates for new DNA synthesis in the second cycle. Therefore, if everything works perfectly, you double the number of copies of your target sequence in each cycle of PCR. You can see from this diagram that double-stranded DNA consisting only of the exact target region to be amplified appears in the third cycle of PCR. You will always have some PCR products with longer pieces of DNA because the original template extended beyond the target region. But starting in cycle 3, the precise target region will grow in abundance until it is soon the major PCR product by far. If everything goes the way it should, after 30 cycles, starting from a single double-stranded template, you will have 2 to the power of 30 copies of the target DNA sequence, which is over 1 billion copies. So, after PCR of our cDNA, you will have many millions of copies of the gene of interest. If you analyze the PCR product by gel electrophoresis, you should see a single band representing the cDNA you want. You can then cut that band out of the gel and extract the DNA from it, giving you a fairly pure sample of your cDNA. This sample can be used in the next step of cloning, which I'll describe in the next video. But before I do that, I want to mention just a few common applications of PCR. One is quantitative PCR, or qPCR, sometimes called real-time PCR. In this method, you include a molecule in the reaction that becomes fluorescent in the presence of double-stranded DNA. So as a particular target sequence is amplified, the fluorescent signal gets stronger. You can measure the fluorescence as PCR proceeds, and from the strength of the signal you can estimate the amount of PCR product made, which should correlate with the amount of template in the original sample. This is another way, besides a northern blot or a microarray, in which you can quantify the amount of a specific mRNA expressed in a tissue of interest. Another application is DNA fingerprinting, which is used in forensics. This method relies on the fact that we all have repetitive regions in our DNA that contain many repeats of short sequences. The number of times the short sequences are repeated can vary from person to person. If you do PCR to amplify many of these repetitive regions, you can see how long each region is in a given individual. The odds that two unrelated people will have many repeated regions of the same length as each other get smaller and smaller as the number of regions you analyze increases. DNA fingerprinting can be used to compare a suspect's DNA with DNA from a sample found at a crime scene, or in applications like paternity tests. A third application is site-directed mutagenesis, used to introduce desired changes into a DNA sequence. In this method, you design a primer that is not completely complementary to the template sequence, but contains one or more mismatches, represented here by the star. As long as the 3' end of the primer will anneal to the template and be used by DNA polymerase in the synthesis step, you will generate new DNA using that primer. That DNA will become the template in subsequent steps, during which the mismatched region will be copied into newly synthesized strands. As PCR progresses, the altered version of the DNA will become the dominant PCR product and can be used in subsequent cloning. Speaking of cloning, in the next video I'll look at how our amplified cDNA can be combined with other DNA to make it ready for introduction into bacteria.